Okay, so hello everyone. Welcome to our today's Zoom seminar. So it's really exciting to welcome our today's speaker, Dr. Amanda Poholek from University of Pittsburgh. So Amanda actually currently is assistant professor from Department of Immunology from University of Pittsburgh. So very brief introduction. Amanda complete her graduate student training in Yale in cell biology, and then she pursued her postdoc training actually in NIH in NIAMS with Dr. John Sher, studying immunology, particular focusing in transcription regulation. So from 2015, Amanda started his independent research program in Department of Pediatrics in University of Pittsburgh School of Medicine. And then all the way up to this year, she actually moved to Department of Immunology as assistant professor. So the main research focus in Amanda's lab is to understand how immune cells integrate signals encountered in the environment to drive functional outcomes at the molecular and epigenetic level in both health and disease. Um, so Amanda really has a long history of training in the field of immunology and in transcription factor control CD4 T cell differentiation. Um, Her lab is investigating the function of this transcription factor BLIMP1 in immune cells in the context of allergic asthma. And then in addition, her lab using the extremely low cell number chip sequence-like assays to profile the epigenetic landscape of T cell isolated directly in vivo to interrogate the epigenome of cells from disease states such as tumors and tissues. Actually, Amanda actually gave a beautiful talk a few months ago in NIH, and I was so impressed by her techniques and also all the fantastic data. So I think it's really worth to share her study with the community. So the general overview, over focus of her research program is to understand how tissue-specific signal change epigenetic and transcription factors pathways to drive new functional program that have important consequence for cell differentiation and function. A few different aspects in Amanda's lab, which mainly interested, including to illustrate that the tumor-mediated contribution to epigenetic landscape of exhausted TCD-CD8 T cells, and then to identify tissue-specific and context-dependent role of BLIMP1 to drive Th2 cells and promote allergic asthma. And then third part is her lab described this BCL6 as the master regulator controlling T follicular helper T cells, development and the requirements for the development of T follicular helper T cells in normal and autoimmune responses. Last but not least, her lab using next-gene sequencing technologies to interrogate immune cell differentiation and function. So with different really groundbreaking discoveries, Amanda will award different honors, including innovation awards in American Lung Association and the Excellence in Research Award in University of Pittsburgh. So I think without further ado, we are very exciting to looking forward to your talk. Thank you so much, Amanda, sharing your study with us today. Please go ahead.
Yeah, thanks so much, Chuan, for that really lovely introduction. And yeah, just to point out, actually, the work I did on BCL6 was my work as a graduate student, which was when I was at Yale. So it was a really fun time for me. For those of you who are listening in on the first 10 minutes of the Zoom call. So today I would like to share a couple stories. One is on the allergic asthma work that we do, and then at the end I'll talk a little bit about some of the cancer stuff that we've been doing with T cell exhaustion. And so all of this work is pretty much published. One aspect of the asthma story is it's still in bioRxiv, but hopefully will be in press soon. And I'm happy to take questions at the end as well. So just to kind of get everybody oriented, I like to start with this slide thinking about T cell function in the context of tissue-specific immunity, and sort of the way that my lab is starting to think about these things. So, of course, we can often think about the tissues as both the site of antigen introduction as well as the endpoint of the inflammatory responses. And I think this is easy to think about in the context of something like a viral infection, right? For example, you get like a respiratory infection, you breathe in the virus, and it's going to start infecting the lung tissue. And then, of course, from there, the antigens from the virus are going to get picked up and travel from dendritic cells that take up that antigen from the lung space into the secondary lymphoid organs that drain that tissue. And in the secondary lymphoid organs is really the site of where your immune system begins to get primed. So this is where your T cells are going to see the antigen presented by the dendritic cell and begin to become activated. And then those cells become effector T cells, getting those signals from the dendritic cell. And then they leave the secondary lymphoid organs, and then they can traffic back to the site of the tissue where they can mediate clearance of the virus, right? And so again, this is very easy to think about in the context of something like an infection, but of course, most of the time we're not necessarily experiencing infections at each of these tissue sites, but rather thinking about how we're really experiencing environmental antigens each day, right? And so the environmental antigens that we are experiencing, that we're breathing in, or eating or touching are distinct at each of these tissue sites. So the things that you're breathing and the things that you're eating and the things that you're touching may not necessarily be the same. And in addition to that, of course, we have a complex and diverse commensal microbiota at each of these tissue sites, and those are distinct across the different tissues. So really, your immune system is constantly having to survey these environmental antigens and maintain tolerance in order to not respond to those things. And so really you can think about the immune system's primary job on a daily basis is to maintain tolerance to the environment and not respond, as opposed to, responding to the infections and the pathogens that they do see occasionally. And so there's, of course, many, many different mechanisms of tolerance, central tolerance in the thymus, as well as things like T regulatory cells, which I'm sure many of you are familiar with. But we also have important mechanisms of tolerance intrinsically in T cells to make sure that the T cells respond and then contract appropriately. And when we lose those intrinsic T cell mechanisms of tolerance, we end up with settings of sort of overt autoimmunity, as highlighted here. And so I kind of want to draw your attention at this point to the transcription factor BLIMP1, which is actually an important transcriptional regulator that was initially identified as a master regulator of plasma cells. So without BLIMP1 in your B cell population, you don't form plasma cells or antibody-producing cells. And although BLIMP is actually expressed in a number of cells, even in the non-immune system, but several years ago, many, many years ago, two separate groups made T cell specific knockouts of BLIMP. And those mice developed a spontaneous multi-organ infiltration and an autoimmune colitis. So you can see here in the colon, this very pronounced inflammation in the colon. And this was really this most severe organ in the loss of BLIMP1 that led to a fatal wasting disease. So what this indicated was that BLIMP was playing an important role in constraining effector T cells that were perhaps being triggered by a commensal microbiota in the absence of any infection, and were important. So BLIMP was important for maintaining autoimmunity, right? Maintaining tolerance to your commensal microbiota. And I think it's important to note that polymorphisms in the human BLIMP1 locus have been associated with some of these T cell mediated autoimmune diseases like IBD and Crohn's. So given this interesting role of BLIMP in potentially constraining T cell mediated systemic autoimmunity, when I first started my lab at Pitt, one of the questions that I was interested in was whether BLIMP and T cells could also control responses to non-infectious stimuli such as allergens, right? Again, thinking about all the different environmental antigens that we experience on a daily basis and need to maintain tolerance to. And so to answer this question, we decided to test the role of BLIMP in a in a situation of allergic response, and in this case, we chose allergic asthma. So allergic asthma is a dominant type 2 T cell mediated disease. It's actually a pervasive disease that's rising both in children and in adults. So it actually has really important clinical implications to understand. Um, and the type 2 cells that are generally driving the disease are Th2 cells. So these are CD4 T cells that can generate type 2 cytokines like IL-4 and IL-13 and IL-5. And it's those cytokines in the lung that drive the lung pathophysiology. So they promote lung inflammation, pulmonary hyperreactivity, goblet cell metaplasia. And these are the things that actually drive many of the symptoms that are associated with allergic asthma, wheezing, lack of breathing, mucus, et cetera. As important as Th2 cells are to the lung pathophysiology, one of the other hallmarks of allergic asthma and of allergy in general is the production of IgE, the antibody IgE. And while initially Th2 cells were considered sort of the cells that drove B cells to make antibodies, we now understand that this is actually the TFH population of cells that is important for mediating allergen-specific IgE and IgE in general, IgG in general, or just antibody production in general. So to test the question that we were interested in about BLIMP1 and allergens, we needed to have a system in place where we could drive allergic asthma in mice, where we had specifically deleted BLIMP1 from the T cells. And so we were fortunate to collaborate with Anuradha Ray at Pitt, who's an expert in type 2 mediated allergic responses, and in particular in allergic asthma. And she helped us set up this chronic inhaled house dust mite model in the lab. And so this was first set up actually by myself and my first technician, Angela Hettinga. And then I recruited my first postdoc, Kun Hee, and he really took over this project and really got this up and running in my lab. And so the system is essentially where you take house dust mite and you immunize the mice intranasally daily for 10 days in a sensitization phase, followed by a rest and a re-challenge, and then a rest and a re-challenge. And when you do this in a control animal, so a mouse that has BLIMP1 intact, you get this beautiful lymphocytic infiltration into the lungs, as shown here with H&E with PAS staining, and this pink staining here in the airways is an indication of the mucus that is forming in the presence of these type 2 cytokines. And so our hypothesis is that BLIMP is constraining autoimmunity. BLIMP is important for constraining effector responses. And so our hypothesis was that in the loss of BLIMP1 in our T cells and in the setting of allergic asthma, the disease would be much worse because we would have increased effector T cell function. But in fact, we actually got a surprise, which is always the best type of scientific experiments that you do. And we actually saw no disease at all in the mice. The lungs look completely cleaned, no allergic asthma, no mucus, no nothing. And to cut a long story short, because this was published several years ago, it turned out that this was because there was a complete absence of Th2 cells in the lung. So you don't have Th2 cells in the lung, you're not able to drive all of those pathophysiological settings of disease and allergic asthma. And so because this work was published, I'm just going to summarize sort of the findings of that first work. So through several experiments, we were able to show that BLIMP1 was actually intrinsically required in the T cells for Th2 development. However, TFH cells and IgE remained intact. So again, you have allergy and IgE, that was completely normal. However, we had a complete loss of the Th2 cells. So BLIMP was sort of dissecting these two pathways between the TFHs and the Th2 cells. And molecularly, the way this was working is that BLIMP is a transcriptional repressor, and it represses a target gene, BCL6. BCL6 is also a repressor, and it can repress GATA3, the master regulator of Th2 cells. So when we deleted BLIMP1, what we had was a huge increase in BCL6 expression, and this suppressed GATA3 and suppressed the formation of Th2 cells. And by deleting both BCL6 and BLIMP1 together, we could restore Th2 cells and restore allergic asthma in this system. So this was a lovely paper, my first big paper from my lab. It was very exciting. But the results really bothered me because as a postdoc in John O'Shea's lab, I had been working on BLIMP and T cells in the context of Th2 cells, and I had done very standard in vitro assays to make Th2 cells in vitro. And I knew very well that you can make Th2 cells with BLIMP1 just fine in vitro. So it's not as if we've overlooked this important master regulator of Th2 cells for 2 decades, right? We also had done an experiment where we had immunized mice with a soluble worm antigen from the schistosome mansoni worm. So worms, of course, also drive pronounced type 2 immunity. And so when we did that and then looked in the draining lymph node, we actually saw an increase in the number of Th2 cells and the loss of BLIMP1, again, sort of along that first phenotype, that in some cases, BLIMP1 is constraining Th2 cells, and in other cases, it seems to be required. And so we really were trying to understand the discrepancy between these two pieces of data. And so we thought, okay, well, what's the difference between the two experiments we've done here? One is the antigen is different, soluble egg antigen from the worm versus house dust mite. And the other one is the mechanism by which we're immunizing. In one case, we're giving it in the foot pad subcutaneously, and in the other one, we're doing this intranasal immunization. So we just did a very simple experiment where we took our two antigens, house dust mite and egg antigen from the worms, and we gave them intranasally, either in wild type or knockouts. And in both cases, in the absence of BLIMP1, we were unable to form Th2 cells in the lung. In contrast, if we took the house dust mite and gave it subcutaneously, now we had a constraining phenotype. So BLIMP1 constrained effector responses when you gave antigen in the foot pad or in the flank subcutaneously. So really this came down to the root of immunization. Somehow when you breathe in the antigen, you need BLIMP1 to form a Th2 cell. So this was where we really wanted to dive into the details of how this response was occurring. How exactly is it that this is happening? So in order to do that, we knew we were going to need some important tools. And we were very fortunate that Bart Lambrick had made a T-cell transgenic mouse that called the Wonder Mouse, where the TCR is specific to DRP1, which is the major antigen peptide in house dust mite. And so we were lucky that Bart was willing to share these mice with us, and we crossed them to congenic markers as well as to a reporter of BLIMP using a YFP reporter so that we could track both the expression of BLIMP1 in our cells in our antigen-specific population, as well as have an allergen-specific population to track by adoptive transfer. And so we started doing our sensitization phase intranasally after 10 days, and then over time we tracked the response in either the mediastinal lymph node or in the lung. And so you can see here that at 72 hours, we can just begin to see in the lymph node the early formation of the TFHs as expressing CXCR5 versus PD1. And by 10 days, the TFHs are the predominant population. And as expected, BLIMP1, as shown here by the YFP, was predominantly in the effector population and not in the TFH population. We saw a little bit of BLIMP1 as early as 72 hours. It peaked at 5 days and then went away, but it was a very small population. In contrast, in the lung, by 5 days, we had this huge burst of BLIMP1 expression, suggesting that once the cells were getting into the lung is when we were really getting the BLIMP1 expression. And so we first wanted to ask what the signals were that might promote BLIMP in the lung. So we had some hints from this from our first paper, long story short, but we had kind of had this idea that IL-10 was a strong driver of BLIMP1. And so we looked at the loss of IL-10 receptor on our T cells. And indeed, in this system, we lost both Th2 cells as well as BLIMP1. So we next wanted to ask, well, what is the source of the IL-10? So we got IL-10 flox mice and we crossed them to a number of cell type specific Cres. And I'm not going to take you through all the negative data, but it turned out that it really was IL-10 from the T cells itself that was important to form Th2 cells in the context of this house dust mite immunization. So the last thing we wanted to do is ask, well, which T cells are making the IL-10 that's driving the BLIMP1 and the Th2 cell formation? And so to do that, we actually did the simplest experiment first, which was to take our antigen-specific cells and transfer them into an IL-10 deficient host or a wild type mouse. Now, in this setting, only the cells that we've transferred in have the ability to make IL-10. And then we immunized 5 days and looked in the lung, and indeed we saw completely normal responses, suggesting that as long as your allergen-specific T cells can make IL-10, that's sufficient to drive BLIMP1 and to drive Th2 cells. So this was lovely, and we had a nice idea, okay, IL-10 is driving BLIMP1 and driving Th2 cells. But we really wanted to demonstrate definitively when this was important, although we saw very little BLIMP at the beginning of the response and more of the BLIMP later in the response in the lung. We really wanted to pin this down. So in order to do that, we crossed our BLIMP1 floxed mice to a tamoxifen-inducible Cre in T cells so that we could induce the deletion of BLIMP1 throughout the course of our immunization. And so we set up several tamoxifen dosing strategies where we're deleting BLIMP either very early in the response or later on in the response after priming or halfway through priming. And I'll just kind of walk through this data a little bit slowly. This is all of our controls that either get vehicle or don't have Cre. And so you get normal Th2 responses in this setting. This is sort of our positive control for no Th2 cells, which is a constitutive CD4 Cre crossed to BLIMP1 floxed. And if you start by looking at the settings and where we delete BLIMP1 late in the response, so after 5 days, we actually didn't really see any difference at all. This is again where we see the most BLIMP1 expression, but we didn't really see any impact of its loss later in the response. In contrast, the earlier that we deleted BLIMP1, that was when we started to see the loss of Th2 cells. So although we saw very few BLIMP positive cells in the lymph node early on, it seemed like this was the critical point at which you needed BLIMP to drive this Th2 response. And very nicely, we saw a corresponding increase in BCL6 as we deleted BLIMP1 earlier and earlier. So we had to start thinking about very, very early signals that would be driving this discrepancy between BLIMP1 and BCL6, much earlier than even what we saw in the lung with IL-10. And so we came back to this really lovely paper from Casey Weaver many years ago that showed that IL-2 signaling was sort of the earliest signal that could differentiate between the TFHs and the non-TFH cells. And of course, IL-2 STAT5 signaling has been shown to promote Th2 cells for many, many years, work from Bill Paul, Jeff Su, Lauren Leonard, and others. And then, of course, we know that IL-2 and STAT5 are also potent repressors of the TFH cells. So we thought perhaps this is actually the earliest signal that's driving that initial BLIMP1 expression that's making a significant difference in actually promoting this Th2 response early on. So to test this, we started by asking, okay, can we start by seeing IL-2 receptor expression and just seeing how that correlates with BLIMP? And so we looked at the very earliest time points after immunization, 24, 48, and 72 hours. You can see here, even before the cells have begun to proliferate. So this is looking at cell trace violet. So the cells begin to proliferate as they become activated. At 24 hours before they've even proliferated, you can see about a third of the cells have already upregulated IL-2 receptor alpha. And then by 72 hours, the T effector population has a small but significant population of IL-2 receptor alpha positive cells. And these IL-2 receptor alpha positive cells correlate very nicely with the cells that express BLIMP1, as well as with the cells that express GATA3. So not all GATA3 positive cells are BLIMP1 positive, but almost all BLIMP1 positive cells are GATA3 positive and IL-2 receptor alpha positive. And then next, we wanted to look at the phosphostat5 phosphorylation, which is the signal that's active downstream of IL-2, and compare that to STAT6, which is the signals downstream of IL-4, which is sort of the predominant cytokine thought to drive Th2 cells. And we could see phosphostat5 as early as 24 hours and then peaking at 48 and then waning, whereas STAT6 doesn't come up until 48 hours, suggesting that this IL-2 signal is preceding IL-4 and STAT6 in the Th2 differentiation. So we drew ourselves a very simple model. IL-2 activates STAT5. STAT5 has been shown to repress BCL6. BCL6 can repress BLIMP1, and then, of course, indirectly suppress GATA3. So to test this, we used a system where we could delete the STAT5 gene using CRISPR in our naive allergen-specific cells prior to adoptive transfer. So now only the adoptive transferred cells have deleted STAT5. And then we did a very short time course of house dust mite, looking at these very early time points after activation. And indeed, when we deleted STAT5, we saw that corresponding increase in BCL6, as we expected, suggesting STAT5 represses BCL6. We saw a decrease in GATA3 expression, as expected, and we saw a pronounced decrease in BLIMP1 expression. So that was great. Our model was holding. And so we decided next to go one step down and delete BCL6. And so again, we did that using the same system, using CRISPR directly into the adoptively transferred cells, the wonder cells prior to transfer. And surprisingly, we didn't see any difference in BLIMP expression, which was a bit of a surprise to us. And we also didn't see really much of a difference in, we didn't see a difference in BLIMP1. However, we did see the expected increase in GATA3, suggesting that it was playing a role in suppressing GATA3. So this made us ask the question, is there another repressor of BLIMP1? Is there some part of this model that we haven't drawn correctly? And we were reminded of some older work that BOC2, another transcriptional repressor, can also repress BLIMP1. And it's been shown to be operative in CD8 T cells as well as in Tregs. And so we asked whether deleting BOC2 would now have an impact on BLIMP1 expression. And so again, we used our CRISPR system to delete BOC2 in our allergen-specific cells prior to transfer. And when we immunized the mice, now we could see this huge increase in BLIMP1 expression, suggesting that BOC2 is actually a primary repressor of BLIMP1. And then very nicely, we were able to use publicly available STAT5 ChIP-seq data, where we could see that STAT5 is binding to BCL6 and acting as a repressor. It's also binding to BOC2, suggesting that STAT5 is really upstream of both of these transcriptional repressors. So this, all this work kind of collectively together allowed us to draw the following model: that IL-2 and activate STAT5. STAT5 can both repress BOC2 and BCL6, and then BOC2 is repressing BLIMP1 while BCL6 is repressing GATA3. And once BLIMP1 is up, you can now repress BCL6 and indirectly allow for GATA3 expression and the formation of Th2 cells. And so this was like really satisfying to work this out molecularly, one little molecule at a time. But we were really curious to try and understand this in the context of the lymph node and in the context of the antigen draining to the lymph node, and thinking about how at this very early stage these T cells could be getting these signals in order to make this decision between IL-2 positive cells getting this signal, becoming BLIMP1 positive, or not getting IL-2 and then becoming a TFH cell. And so to answer those questions, we turned to spatial transcriptomics to try to identify in situ in the lymph node where this might be occurring. And so we did this work actually quite a while ago. So we started by using some of the earliest 10x Genomics Visium platforms, where the resolution is nowhere near single cell. It's lots of cells for each dot. And so just to kind of orient you, what we're looking at here is a immunofluorescence stain, just to sort of hone in on the T cell zones and the B cell zones here on the left. And then overlaid on top of that, we have our spatial transcriptomics data, where each of these circles is an individually barcoded spot that probably has somewhere in the order of 20 to 25 or 30 cells per spot. These are 55 microns in diameter. And we can sort of look within each of these spots for transcriptomes that are associated with cell types of interest, like TFH cells or Th2 cells or B cells. And the heat of the spot or the color of the spot is an indication of the percentage of that spot that's associated with that transcriptome. So I think the B cells is kind of the easiest one to look at because it's easy to find the B cell zone. And you can see that the transcriptomes of those spots that are overlaying the B cell zone have very high, correlation to B cell transcriptome. So that makes sense. So if we look here, the top row is a day 3 lymph node, the bottom row is a day 5 lymph node. And you can see here at day 3, the TFH transcriptomes are lining up sort of in the T cell, B cell border and the interfollicular zones at day 3, but by day 5, they've completely switched into the B cell follicle, which is in line with how we understand TFH cells to work. They initially get a signal at the TB border, and then they move into the B cell follicle where they interact with B cells. So nothing we didn't already know, but very nice to see as well. Interestingly, the Th2 cells kind of start a little bit deeper into the T cell zone, and then also by day 5 switch into this T cell, B cell border zone into the interfollicular zones. And so because at day 3 we sort of had this slight change in localization, and certainly one by day 5, we had this idea in our mind that perhaps there was a spatial localization that was underlying this IL-2 driven difference. And so to really try to understand that, we collaborated with a computational biologist in the Center for Systems Immunology, Dr. Jishnu Das at Pitt. And he had recently developed an algorithm called SLIDE, Significant Latent Factor Interaction Discovery and Exploration. And SLIDE is an interpretable machine learning technique for identifying sort of significant latent factors that underlie outcomes of interest using high-dimensional omic data sets. Okay, and so what that means is you have a high dimensional omic data set with all of these observable features, and you have an outcome or response of interest, which we call Y. And what SLIDE does is it utilizes these features to identify sort of hidden latent variables. And so a latent variable is something that you can't see but is there and driving the response or contributing to the response. And so importantly, this algorithm really provides inference beyond prediction. And this work was published in Nature's Methods recently, so I urge you to go look it up, and it's all open source if you're interested in using it. And so the output of SLIDE kind of looks like this, where you get these latent factors that are filled with genes. Each of the latent factors has genes, and the genes are assigned by the algorithm to be either associated with one of the two observable features. Now, in our case, we were interested in the spatial localization of Th2s versus TFHs. changes and what might be a hidden latent variable that underlies that. And so you can see here, for example, Z12 is one of our latent factors, and it only has genes that were associated with Th2 cells, and they're all associated with cell adhesion and migration, giving the idea that even at day 3, the Th2 cells are starting to already prime themselves to leave the lymph node and go back to the lung. Z13 was the one we got really excited about because although most of the genes in Z13 were TFH associated and associated with B cells, which again makes sense because that's what TFHs do, they interact with B cells, we also had this IL-2 receptor alpha come up as a Th2 associated gene, giving us the idea that perhaps the IL-2 signaling itself had a spatial component to it to drive this response. And then looking at the correlation network within Z13, we could see that IL-2 receptor alpha was a strong node and had high positive correlation to many of the other genes within our latent factor. So in order to test that, we actually decided to look for IL-2 in situ in the tissue. So it's very hard to stain for cytokines at the protein level. So we started by using RNAscope, which detects IL-2 message at the RNA level. And each of these purple spots is an individual IL-2 positive message that we could find in one of our tissue slices. And then what we did was we decided to draw a 50 micron radius circle around each of the IL-2 positive spots and then count within that circle how many of the cells nearby only had GATA3, only had BLIMP1, or had all of the three together, right? And so that's kind of shown here quantitatively. And what we found was that around the IL-2 positive spots, we actually had an increase in cells, in T cells that had both BLIMP1 and IL-2 receptor alpha and GATA3, suggesting that IL-2 was sort of a micro niche where you had these nascent Th2 cells that were BLIMP1 positive beginning to form. In order to better understand this and have better tools, we were able to get IL-2 reporter mice from KZ Weaver. And you can use these mice to find IL-2 positive cells, as shown here. And then around those IL-2 positive cells, we were able to find BLIMP1 positive cells, IL-2 receptor alpha positive cells, as well as GATA3 positive cells. And then if we actually go ahead and delete IL-2 receptor alpha from our allergen-specific cells, not only is the BLIMP1 expression completely lost, but we also lose the expression of the GATA3 surrounding those microniches. And if you quantitate that, it's shown here that you can find these IL-2 positive cells. And again, the distance of those cells is much shorter to the cells that express GATA3, IL-2 receptor alpha, and BLIMP1, as opposed to just GATA3 alone. So really indicating that these are sort of important spatial microniches. So what are these cells? Obviously IL-2 can be made by a number of different cells, but it's predominantly CD4 T cells. We also looked at the spatial localization of these IL-2 positive CD4 T cells within the lymph node, and they're predominantly found in the T cell B cell border region, as well as in the T cell center, but not so much in the B cell zone. So kind of to sum up this first part, what we think we've shown are the really earliest signals for forming a Th2 cell in response to inhaled allergen, and really describing the molecular pathways that form the cells that go on to promote allergic asthma. So our model at this point is you breathe in an allergen that's taken up by migratory CDC2s in the lung. They go to the lymph node where they prime T cells specific for the allergen. At the very earliest stages, some of those cells localize nearby an IL-2 positive CD4 T cell, and they receive these very early IL-2 signals through STAT5 and through repression of both BCL6 and BOC2. They're able to upregulate BLIMP1, and that starts this initial response to start driving GATA3. At some point, these cells begin to make IL-10, and then the IL-10 actually forms an autocrine-paracrine feedback loop to further enforce BLIMP1 as the cells migrate out into the lung, where they really burst both IL-10 and BLIMP1 expression, and are the Th2 cells that really go to the lung and drive allergic asthma. And we think these spatial microniches of IL-2 really support the initiation of these BLIMP-positive Th2 cells. So I want to totally shift gears now to another part of the lab and a completely different set of work that we're doing, not on CD4 T cells, but on CD8 T cells, and not in allergic asthma, but in the context of solid tumors. And so this is in part because when I got to Pitt, so many people at Pitt, we have a really, really strong cancer immunology group there. And so I had not really been exposed to cancer immunology prior to coming to Pitt. I certainly knew a little bit about it, but I hadn't really been around people really actively working on it at the highest levels. Um, and it was interesting to start talking to them and to really start thinking about the tumor as this like very unique and interesting tissue space where the immune cells are going and have to respond to a very different environment. And in particular, I started talking to Greg Delgoff, who has a very strong interest in T cells and in the metabolism of T cells in the tumor. And we both were really interested in just how do T cells sense different signals? How do they respond? How do they do different things? And so he kind of oriented me to understanding, how interesting the tumor microenvironment is with these really niches within the tumor itself, where the cells in the tumor are sort of competing for resources, right? And so you have these hypoxic regions, for example, that have increased lactate and limited glucose, and changes, of course, in oxygen availability. And so the cells that are within those hypoxic regions actually look distinct from the cells that are not in the hypoxic regions. And of course, we now understand that the CD8 T cells that go into the tumor undergo this very interesting and distinct differentiation pathway of T cell exhaustion. So normally CD8 T cells during an acute infection differentiate to effector cells, and then once the antigen is cleared, they become memory cells and are, hang out in case they ever see the infection again. However, in settings of chronic antigen, either in a chronic viral infection or in the case of tumors, the cells undergo this alternative differentiation towards exhaustion, which we now understand to both be a very heterogeneous state as well as a progressive state. So you have cells in the tumor that exhibit sort of some effector function, and they, and most importantly, have a lot of stem-like factors and can maintain some self-renewal properties, and we call these sort of progenitor exhausted cells. These cells under settings of immunocheckpoint blockade can differentiate into effector-like cells that actually can drive some tumor clearance. However, they also, in the setting where they don't get some sort of help from immunocheckpoint blockade, undergo a progressive process towards this terminal exhaustion where they become progressively dysfunctional, increase inhibitory receptor expression, lose effector cytokines, lose metabolic function. And to me, what's interesting about this is just that this is a very distinct T cell differentiation pathway, and that what underlies this pathway are very distinct transcription factor profiles, right? So these progenitor cells predominantly express TCF1 as a transcription factor, whereas these terminally exhausted cells express both BLIMP1 and TOX. And when you have two cells that express, different transcription factors in such pronounced ways, that usually is an indication that they have distinct transcriptomes, which we know, of course, know after many groups have done different RNA-seq experiments and have definitively shown that these cells have different transcriptional profiles as well. And so given that they have different transcriptional profiles, what we were really interested in understanding was whether they also had distinct epigenetic profiles that underlied those transcriptomes that were different. So this work was a huge collaborative effort with Greg Delgoff's lab. It started with a very talented undergrad in my lab, Natalie Rittenhouse. And then when she left to go to graduate school, it transitioned to my first graduate student, Rhodes Ford, who graduated last year and is now a postdoc at UCSF. And in Greg's lab, it started with a very talented graduate student in his lab, Nicole Sharping. And then when Nicole graduated and became a postdoc at UCSD, it transitioned to Paolo Vignali, an MSTP student in Greg's lab. So, this was a big effort over several years. And so we started by using the system that Greg had up and running in his lab, which was a very simple B16 melanoma system where you can inject B16 tumor cells under the skin, and 14 days later you get these lovely tumors with beautiful exhausted T cells in them. And if you do flow on those tumors and look at the CD8 T cells, you can profile them in just using two inhibitory receptors, TIM3 and PD1. And the cells that express no TIM3 and very low levels of PD1 are these progenitor cells, and these terminally exhausted cells express both high levels of PD1 and TIM3. And we could sort those cells out at the bulk level and do RNA-seq, and we could see that they were, again, very transcriptionally different, the PD1 lows versus the PD1 high, TIM3 double positive. And when we looked at specific genes that we all kind of know and love as signatures of the progenitor fate or the terminal fate, we had the same profile. So basically, we had a system that was consistent with other people's designations. And so we were really interested in understanding the epigenome. And when Greg and I actually started talking talking about this project, it was probably like 2016, 2017. And we were trying to come up with a way to do the epigenome with low input ChIP-seq. And it was really challenging to get enough cells. And then we were very fortunate when Steve Henikoff published this new assay called cut and run, which is basically a low cell number ChIP-seq-like assay that can be done on very, very few cells, under 100,000 cells. And it really uses this wonderful covalent protein A complex to MNase. And then under settings of low temperature and calcium, the MNase is active and can bind and cut DNA. And the protein A can bind to an antibody that you've put into the cell. So it's just like ChIP, where you have an antibody, it binds to your protein of interest, but you're using protein A MNase to actually do the cutting and extracting the DNA. And so this allows you to do this on many, many fewer cells. And so we sort of just took our system, sorted our different populations, and did cut and run for a variety of different epigenetic marks using antibodies to different histone modifications. And so we decided to focus on four histone modifications to start with, three that are sort of associated with open chromatin and positive transcription, K4ME3, which is a mark of active promoters, K27 acetyl, which is a mark of active enhancers, and K9 acetyl, which is a mark of active gene bodies. And then one mark of repressed chromatin or loss of gene transcription, K27 trimethyl, which is a mark of dynamic heterochromatin. Okay, so the first thing we did was just ask the question, if we start looking at the genes that we know are upregulated in our terminally exhausted cells compared to our progenitor cells, and look at the epigenomes that underlie those genes, do we see things that make sense? And very nicely we do. So you have genes that are upregulated in terminally exhausted cells. Upregulated genes usually have open chromatin, so you see an increase in open chromatin marks at each of these genes and a decrease in the repressed chromatin mark at each of these genes as the cells become of that phenotype. And you can see that sort of in an example of that right here at the TOX gene, which is of course much more highly expressed in the terminally exhausted cells. So you can see that you have repressive marks in the PD-1 low cells that go away in the PD-1 high cells around the TOX gene, whereas you have an increase in these three active marks in the terminally exhausted cells for the TOX gene. Okay. So the next thing we wanted to do was start looking at interesting states of chromatin. And one of the ones that we were kind of curious about was this state of chromatin termed bivalency. So bivalent chromatin is a state where you have both active and repressive marks on the same nucleosome, right? And so this is generally considered to be something that you find in a pluripotent or stem cell-like state. And they're usually found at cell type specific transcription factors where your cells are, a stem cell. They are going to receive information to become one state or another state as a stem cell. And having bivalent chromatin at some of the key genes that are going to drive that process makes it a lot faster. You don't have to open the chromatin and turn on the promoter as well as remove repressive marks. You have both. You just remove one, remove the other, and off you go. And so we were kind of curious whether we would see patterns of bivalency associated with these progenitor cells that were indicative of this more pluripotent or stem-like state. So the first thing we had to do was, using our data sets, we had to first identify where was the bivalent chromatin. So to do that, we used a hidden Markov model called Chrome-HMM, which basically allows you to bin all of your chromatin and then assess the marks that are present at each of the bins according to the different marks that you're interested in. And so you can see here in our CD8 TIL, which we sort of bulked all together, we could find one state of chromatin, state 4, where you had both active and repressive marks in the same regions of the chromatin, as opposed to state 2 and 3, which were primarily places with repressed chromatin, or state 5, which was primarily active chromatin. And so then you can take those states of chromatin and you can then annotate them back to the chromatin for each of your individual populations and look at the percentage of the chromatin that falls into each of the states that we've identified. And so we were kind of surprised to find that actually all of the exhausted cells had more bivalent chromatin than an effector cell. And as they became progressively exhausted, the amount of bivalent chromatin actually increased, which was not really what we had expected. And so then we took all of those regions and we very carefully annotated them to TSSs of genes, and then we looked very carefully for the transcriptional amounts of those genes, and then counted up the number of cells that really, the number of loci that really truly met the mark of bivalency. And we found that the terminally exhausted cells had more genes with this bivalent promoters. And many of those genes actually were genes that are important for immune function, things that we know to be important for even terminal exhaustion. So suggesting that somehow this bivalent chromatin was driving gene repression in these terminally exhausted cells. And so we wanted to confirm this because, of course, we're doing sort of bulk assays for epigenetics, and we're looking at places where you have these same two marks in close proximity. So to answer that question, we did a proximity ligation assay in single cells where you have two antibodies that each are binding each of your two marks, and then you use secondary antibodies that have oligos that can be ligated only when they're within a very close distance of 40 nanometers or closer together. You can amplify this and then use these fluorescent probes, which will only bind if these two marks are, again, in that close proximity. So we did this by microscopy where we sorted our cells onto glass slides and then did the PLA assay onto the cells in the glass slides. And I think you can see very nicely that our terminally exhausted cells did indeed have an enrichment of these fluorescent marks or these PLA foci, indicating that this really is true bivalency at the single cell level. So we were sort of curious what would be driving this bivalency. And one of the things we kind of asked early on was, well, is this a feature of exhaustion or is this a feature of tumor-mediated exhaustion? Do you see this in all settings of exhaustion? So to answer that question, we decided to start with an RNA-seq data set that was generated by Nick Haining, where he had actually looked at both progenitor and terminal exhausted populations, both from the B16 tumors as well as from a chronic viral infection, LCMV. And we took that data set, we used our bivalent gene list that we had from our data set, and we found the gene expression in their data for their RNA-seq data. And so very satisfyingly, in their tumor data, those same bivalent genes that we identified had a decrease in expression in the terminally exhausted cells compared to the progenitor cells. But we didn't see any change in expression of those genes in the chronic viral infection, suggesting that this bivalency, this decrease in expression that you see in terminally exhausted cells at these bivalent genes was something that was specific to tumor-mediated exhaustion and not necessarily a feature of all T-cell exhaustion. So around this time, Greg's lab was also really showing very nicely that hypoxia was a driver of exhaustion, of T-cell exhaustion. And as part of that work, he had generated this B16 tumor line that was less hypoxic by deleting NDUFS4, which is a component of complex 1 of the mitochondria. So we had this system where the tumors themselves were less hypoxic, and this meant that the cells were actually more functional. So you had an increase in functionality as well as a decrease in inhibitory receptor expression. So they looked less exhausted when you had less hypoxia. And so we used this system to ask whether we could see changes in bivalent gene expression when our tumors were less hypoxic. And indeed, we found that when we used these less hypoxic tumors, bivalent gene expression in our terminally exhausted cells increased, and we also saw changes by PLA in a reduction of bivalency at these genes. We did cut and run to see which of the two marks was changing, and we found that actually it was the repressive mark that was decreasing when the cells were less hypoxic, while the positive mark of promoters was intact. So really, by decreasing K27 trimethyl, we were removing that bivalent mark and restoring gene expression. And one of the reasons we were kind of looking in this area, and one of the reasons that I was really excited about the idea of hypoxia contributing to this, was because these papers came out in 2019 that showed that the histone demethylase that can remove K27 trimethyl can directly sense oxygen. And in settings of hypoxia, KDM6A becomes dysfunctional and can't remove K27 and leads to increases in bivalency. And so we looked at the expression of these histone demethylases as well as EZH2, the methyltransferase that adds K27-trimethyl. And we were really excited to see that KDM6A doesn't transcriptionally change, although this is the one that can directly sense oxygen, while KDM6B transcriptionally goes way down in our terminally exhausted cells, while EZH2 goes up. And when you change the hypoxic environment, you change the amount of the histone demethylase that's present. So what this suggested to us is we had a system where you're adding too much K27-trimethyl. You're not able to take it away. This is leading to inappropriate K27 expression, trimethyl expression, and bivalent chromatin and reduced gene expression in our terminally exhausted cells. So the last thing that we wanted to do was determine whether overexpressing KDM6B, the one that's transcriptionally controlled, could restore effector function in our terminally exhausted cells. So to do that, we used a B16 OVA system and then OT1 cells that could recognize the B16 OVA, and we overexpressed KDM6B using a retrovirus. And we adoptively transferred those cells after the tumors had begun to form. And then 7 more days later, we were able to isolate those cells and look at the features of the cells that had overexpressed KDM6B. And remarkably, we found that the T cells that overexpressed KDM6B had more interferon gamma, more granzyme B, more TNF, and more IL-2. They also had restored gene expression of some of those key bivalent genes that we had identified, such as BCL6 and SLAMF6. And it's a small difference, but it actually was sufficient to drive slightly smaller tumor sizes after the transfer of the OT1 cells. So proof of principle that we're really increasing anti-tumor immunity by overexpressing KDM6B. However, when we looked at sort of surface marker expression or transcription factor expression that designates the phenotype of the cells, such as PD1, TIM3, CD39, as well as TOX, we didn't see changes in those genes, right? So the bivalent genes can come back, the effector function can come back, but we're not actually changing the terminal differentiation of T cell exhaustion per se. So really what we're doing is we're going in and we're epigenetically restoring function without altering the differentiation of the cells. And so the reason that we think that this is important is because, in general, we sort of now think in the context of the cancer immunotherapy landscape as these terminally exhausted T cells are kind of a waste of space. They're no longer functional. We can't rescue them. We've got to focus on the progenitor cells. And certainly, I think that that's true. There's lots of data out there that suggests that that's true. But I think what our study somewhat suggests is that these terminally exhausted cells still do have potential if you're able to actually change their epigenetic landscape or change them fundamentally in some way. And so we think that normally we have settings of terminally exhausted TIL. I didn't have time to talk about this part of the paper looking at active chromatin states and enhancers, but bivalent chromatin is certainly contributing to gene expression being turned off. And in settings where you can modulate the oxygen environment or restore expression of KDM6B function, you can actually restore gene expression of key effector genes and promote anti-tumor immunity. So I think I'm okay on time for questions still. I really just want to take a moment to thank the people that did the work. I tried to highlight them as I went. All of the allergic asthma data was performed by my first postdoc, Kun Hee, who actually recently left the lab to become a lead research specialist at the Center for Vaccine Discovery and Global Health at the University of Maryland in Baltimore. The work with Greg's lab was done by my first graduate student, Rhodes. And so this is sort of a picture of the lab from a few years ago, and this is actually a very recent picture of the new group. And several of those projects are now being worked on by people in the lab. Obviously, I have had a ton of collaborators that both in the University of Pittsburgh and outside of the University of Pittsburgh that have helped us along the way with either reagents or mice or informatics analysis and intellectual support. I highlighted the collaboration with Greg in particular. And I also want to in particular thank Will McDonald, who is the facility director of the sequencing core that I run, that does all the spatial work, and he was really instrumental in helping us getting Visium up and running, and for my lab and now for everybody else at Pitt. And of course, I have to thank all of my funding sources. And with that, I'm going to stop sharing so that people